Good afternoon, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. This is the second part of our three-part series called New Frontiers in Liquid Biopsy Research, sponsored by Thermo Fisher Scientific. The title of today's webinar is Evaluation of a Pan-Cancer Cell-Free Assay to Meet Unmet Research Needs. Our panelist today is Dr. Luca Quagliata, Senior Director for the Contract R&D Unit at University Hospital Basel's Institute of Medical Genetics and Pathology. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box on the upper right side of the control panel. When you click on Send To, please select All Panelists. We will ask the panelists questions after the presentation has concluded. Dr. Quagliata, please go ahead.
Hi. Good evening, everyone. And first of all, I would like to say thank you very much for the invitation. It is, in fact, a pleasure for me to share with you some of the experiences that we have accumulated in the last couple of months while testing and evaluating the pan-cancer cell-free assay. Before I share with you some of the data that we have collected in my laboratory, this is my speaker disclosure for today. Now, once more, before I start to share some of the data we collected, I would like to put our work in a frame. And the frame that I'm referring to is the one of precision medicine, which really represents a shifting paradigm in medicine today. Here, what I try to do is to summarize the whole concept of precision medicine in a nutshell. Certainly, it is not an exhaustive slide, but what I would like to do is to try to give you an idea of what typically happens in our everyday routine setting. Typically, a patient is showing up at the clinic having a certain clinical suspect. The investigation starts by an imaging approach, meaning performing an X-ray. We eventually found out that this patient has, in fact, or might have a mass in his chest. Then we will proceed with another level of investigation, meaning increasing the level of complexity of our investigation, for example, performing a CT scan. Eventually found out that there's very high chances that indeed this patient has a lung tumor. Typically what happens next is a surgery room is set up, and as you might clearly imagine, this requires yet another level of complexity, and a tissue sample is collected and is sent to an institute of pathology where a trained pathologist will revise this case, eventually telling you that, yes, unfortunately, this patient has an adenocarcinoma of the lung. Now, as a matter of fact, in the year 2018, there are out there a number of possible treatments for this patient. But the question is, which one is the most appropriate? Which treatment is the most appropriate based on the molecular characteristic of this tumor? In order to answer to this question, what typically happens is that DNA or RNA are extracted from tumor cells of these samples, and they are profiled. So a molecular profiling of this patient will be generated, typically by NGS. Next, the generated data will be analyzed and matched with the clinical information of this patient to wrap up a medical report. This report is then offered to the oncologist who is eventually picking up the most appropriate treatment for this patient. Now, additionally to this, in the last couple of years, something else has happened. In fact, therapy assessment and disease monitoring has been an integrated part of our work at an Institute of Pathology. In fact, what typically happens is that after the first treatment, the patient comes back to the hospital in order to receive a constant monitoring. And this is typically happening by PET scan, but more recently also liquid biopsy has been used in order to assess, for instance, the presence of resistance to certain kind of treatments, or for example to looking at the variation from a molecular point of view in terms of genomic landscape of the tumor. Now, based on this additional information, a new treatment decision might be eventually taken by the treating physician. So what I try to do in here, once again, is to summarize the whole concept of precision medicine into a nutshell. And the way I present it to you, it seems like it's a very linear process. However, as you might clearly imagine, this is a fairly complicated process because it requires the interaction of many different professionals, starting from the general physician, moving on to the radiologist, eventually the people working in the surgery room, the pathologist, the molecular pathologist, as well as the molecular biologist and laboratory technician, and back again, the oncologist, not to forget the patient as well. So it's a fairly complicated process. What I also want to mention is that performing comprehensive molecular diagnostic is not something that we should see as happening in the future, but it's something that is already happening today in our laboratories. In fact, if you look at this slide here, those are the most recent testing guidelines that the National Comprehensive Cancer Network, one of the major stakeholders in the United States, giving guidelines to treating physicians, has released about non-small cell lung cancer in advanced stage of disease, meaning in the metastatic setting. What the NCCN network is suggesting at the moment is that on the top of the classical driver mutation testing, and I'm referring in this case of non-small cell lung cancer to EGFR, KRAS, BRAF, and HER2, as well as ALK and ROS, testing should be conducted as a part of a broad molecular profiling, suggesting that approach such as the one offered by NGS should be used in order to analyze those samples. Now, as a turn back, this is generating quite a consistent pressure, so to say, on the molecular diagnostic lab as the one we're having here in Basel. This is just an overview about the number of samples that the molecular diagnostic unit here at our institution in Basel has analyzed over the last couple of years. And as you can see, in the last year, we have been always growing in the number of samples analyzed using complex molecular profiling with an average growth in the last 8 years of about 10% every year. Now, I also want to mention that by doing this, we are piling up quite a consistent number of samples. And here in Basel, for example, we have now more than 10,000 patients that have received so far molecular profiling, the vast majority of them by using NGS. Another very important thing that I would like to mention, and this will allow me to reconnect with the concept of liquid biopsy, is that we have seen really a radical change in the type of samples that we are analyzing in our activity. Indeed, in the past, in the molecular diagnostic unit, we were used to analyze only resected specimen, meaning that we were able to provide molecular profiling and molecular assay information only starting from a consistent amount of starting tissue. Nowadays, this is really changing. Our routine activity is really changing, and in fact, the vast majority of our samples are constituted by either small biopsy or cytological specimen. What I also foresee for the future is that there will be a continuous shift also in direction of liquid biopsy, which are nowadays making their way through the clinic. Now, it is important to mention that each of these different type of tumors comes with different problems and also comes with different extraction methods as well as different tips and tricks to generate specific library, and I'm of course referring to solid tumor versus liquid biopsy, as well as with different bioinformatic pipelines in order to analyze data and generate meaningful NGS profiling. Now, as I mentioned, there's a number of new guidelines suggesting that liquid biopsy should be used to the testing palette, for instance, in the context of non-small cell lung cancer. However, I have to say that the major stakeholders, such as College of American Pathologists, IMP, American Association for Molecular Pathology, as well as the European Society of Pathology, have not yet released full guidelines when it comes to liquid biopsy. There has been some suggestion along to the classical guidelines which have been introduced late last year for NGS, but not clear guidelines on how, when, and what is the most appropriate way to perform liquid biopsy as a routine test in a clinical setting has been so far released. Nonetheless, clinical liquid biopsy, at least for some special application, already made their way to the clinic. Now, question is, why liquid biopsy? Why are we all talking about liquid biopsy in the last couple of years? Why there has been such a huge interest in using liquid specimen? First of all, I think we should start by defining terms. Liquid biopsy can actually refer to sampling of known solid biological tissue, which most commonly refer to blood, but actually also refer to saliva, urine, as well as cerebrospinal fluid. Now, in this seminar, we will mostly focus our attention on blood-related application because those are the ones, in fact, that are most typically used in a clinical setting. Now, I also like to point out some of the advantages, but also the limitation that we have in having such an approach, meaning using liquid biopsy. In fact, one should not be fooled now that by using liquid biopsy, we could sort out all the problems that we need to face in our routine activity. Nonetheless, there are certainly some important advantages in using such an approach. For instance, while using solid tumor, we do have a major problem talking about or referring to invasiveness of collecting a tissue biopsy, or for example, having a lot of limitation in the sites and the tissue sampling that we can collect by using a solid tumor biopsy. On the other hand, what we have by using a liquid biopsy is a number of advantages, such as, for instance, being less invasive, being also actually less expensive, and also reducing the turnaround time in order to deliver back result generated by the analysis of liquid biopsy associated tests, as well as also being able to possibly explore tumor heterogeneity. Now, am I somehow suggesting that a new chapter in the molecular diagnostic book is actually needed? In other words, is liquid biopsy going to change our routine activity in the molecular diagnostic lab? Well, there's still a long way to go, and today with you, I would like to share some of the initial steps that we are moving to and some of the paths that we have been walking in the last couple of months in order to write this new chapter. Once again, a bit of terminology and a little bit more of introduction. Now, when we are referring to a liquid biopsy, once again using blood as reference samples, there are three main components that we are interested to. Those are circulating tumor cells, cell-free DNA, and in general cell-free nucleic acid, it could also be RNA, as well as exosome. For the purpose of this talk today and this presentation, I will be exclusively focusing on cell-free DNA. This because it is so far the only component out of the one making up in liquid biopsy screening testing today that made somehow its way to a routine clinical setting. The other one holds a lot of promises, but today they are not yet used as a routine test. Now, going back to cell-free DNA, typically we found something in between 2 and 40 nanogram of circulating cell-free DNA per ml of plasma on average in advanced cancer research samples. I really would like to point out that this amount of cell-free DNA can be extremely variable and is influenced by a number of parameters that are directly related to the status of the patient and to the stage of the disease of the patient. Indeed, it is pretty well known nowadays that patients that are in a more advanced stage of disease are typically presenting with a higher level of cell-free DNA, while patients that are in an early stage of disease are typically presenting with lower level of circulating cell-free DNA, which makes some of its application extremely challenging in those kind of patients. Another main reason why we are talking about liquid biopsy in the last couple of years is related to the problem of tumor heterogeneity. As we all know, tumor heterogeneity represents one of the major limitations in nowadays precision medicine, at least the way we know it. And liquid biopsy might actually help us to tackle some of these problems related to tumor heterogeneity. But what tumor heterogeneity indeed represent in a real clinical setting? Typically, as you can see from these slides, we are only receiving a single biopsy from a tumor. Now, this single biopsy might have been taken in a part of the tumor that could have a unique genetic makeup that might not represent the complexity and the entire landscape of the tumor mass at all. So this meaning that there might be some clonal mutation that we might be missing by simply taking or analyzing a single tumor biopsy. Now, given the fact that multiple biopsy is something that we cannot go for a number of medical, ethical, as well as economical implications, a liquid biopsy might eventually help us to partially undertake this problem. And in order to demonstrate that tumor heterogeneity is not something that it's just on the paper, but it represents, in fact, a true clinical problem, here I would like to share with you one clinical example. This was a patient that presented at our clinic having an adenocarcinoma of the lung, as represented by this first tissue biopsy, where we performed a classical sequencing, especially looking at EGFR exon 19 deletion, which are typically alterations in non-small cell lung cancer patients. And in this very specific case, we couldn't find any of these genomic alteration. Thus, in this case, patients are not eligible with classical tyrosine kinase inhibitor and unfortunately have a relatively bad prognosis. What happens is that we received a second biopsy from the very same patient, and now, as you can see here, also without being a histopathologist, you could easily recognize that the morphological path in the first biopsy is very different from the one that we could observe in the second one. Indeed, this patient had a mixed adenosquamous carcinoma of the lung. And most interestingly for me, that I'm not an histopathologist, but I'm working on the molecular side of it, is that by sequencing the second biopsy, so the one coming from this part of the tumor, we could actually observe that it harbored an EGFR exon 19 deletion. Now, based on this information, the patient could be treated with a tyrosine kinase inhibitor and has a completely different outlook in terms of prognosis. Now, as I mentioned before, in the vast majority of cases, unfortunately, we don't have this second biopsy, and this is still how tumor heterogeneity might limit the usage of precision medicine as we know it today. So, using liquid biopsy, as I already mentioned before, might in fact have a very real impact, helping us to partially undertake this problem. In other words, the clinical issue that we have today is that despite the very impressive initial response, most patients that are receiving first-generation EGFR tyrosine kinase inhibitors, and I'm talking once again in the context, mostly in the context of non-small cell lung cancer, they will develop some kind of resistance between 10 and 16 months after treatment. Now, in the vast majority of cases, acquired resistance to tyrosine kinase inhibitor is associated to mutation in EGFR that are happening in the position known as T790M. Important to mention, this mutation can be tested and can be assessed by using liquid biopsy. Indeed, in a routine setting, after treatment with tyrosine kinase inhibitors, we will monitor the patient by performing liquid biopsy several times after treatment to eventually look for the appearance of such resistance mutation, which will allow the clinician to put the patient on a new kind of treatment, also known as third-generation tyrosine kinase inhibitor, therefore allowing the patient to have an additional clinical option to fight against this disease. Now, this is one example, this is one clear clinical example of liquid biopsy application in a routine setting. However, I have to say that despite a lot of interest in the field, such number of application is very limited at the moment. Indeed, before I'm going to show you some of other possible liquid biopsy application, I would like once again to point out a few very important points that are necessary to be tackled before liquid biopsy will be fully used in a clinical setting. Indeed, things that are extremely important to be assessed are analytical validity, clinical validity, as well as clinical utility of a test. And for liquid biopsy, while some of these points have been already addressed, some other of those points still need open and needs to be proven, needs to be, in fact, demonstrated. Now, in this very specific talk today, for the sake of time, I won't be able to properly focus on clinical validity as well as on clinical utility. However, we will be able to discuss together about analytical validity. And for analytical validity, I'm referring to the ability of a test to properly and reliably detect variants of interest. And of course, this refers to the measure of accuracy, sensitivity, specificity, and robustness of a certain assay. So as I mentioned before, this is something that has been already demonstrated for several applications for liquid biopsy. However, clinical validity as well as clinical utility has been not fully documented and demonstrated for liquid biopsy. Now, when we're talking about cell-free DNA in the context of liquid biopsy, I mentioned before to you that the amount of that cell-free DNA can be very variable. But typically, in 10 ml of blood, we might ending up having about 20, actually 5 to 40, on average 20 nanogram of cell-free DNA, which would typically correspond to about 6,000 genome copies, which would correspond again to 6 mutant molecules with about allelic frequency or electromolecular frequency of about 0.1% typically. Now, as you might understand, in order to be able to look at mutation at such low allelic frequency, we need to push the limit of detection of the assay that we want to use to tackle such kind of mutation down to extremely low level. And indeed, one of the major limitations for using liquid biopsy is the necessity to be able to go down to extremely low allelic frequency. Another very important point that I would like to mention is that cell-free DNA won't be the solution for all different tumor types. Indeed, there are several tumor types that are known to shed quite a consistent amount of cell-free DNA in the blood. I am referring, for example, to bladder, colorectal cancer, ovarian, pancreatic, breast, melanoma, hepatocellular carcinoma tumors. However, there are also some tumor types for which the use of liquid biopsy might be limited per se by the fact that the anatomical location of the tumor or the intrinsic poverty of the tumor are associated to a very limited release of cell-free DNA in the circulation. And in this case, I'm referring to tumors such as renal cell carcinoma, thyroid, and glioma. In those cases, even having extremely highly sensitive method won't be enough to detect variants for such kind of tumor. However, for the other one that I mentioned before, we do have nowadays technology that allow us to go down to sensitivity and limit of detection that are lower than 1% when referring to allelic frequency. And the different spectrum of technology that I'm referring to is here depicted in this slide, and I would like to mention as a first thing that this slide is a courtesy of Professor José Costa from Porto. Indeed, what you see in here as all the different technological approach that we could use to analyze cell-free DNA. And you have here on one axis relative sensitivity of the different assays used compared to the different breadth of the assay used, meaning to the number of bases that you could address by using this different assay. And what you see in here that in between using an exon sequencing approach and the beaming digital technology, we have NGS. NGS, in fact, has been now recently used in a number of different applications when analyzing cell-free DNA, and it allows a very good flexibility in terms of level of targets that can be assessed at the same time, and also allows you to go down to limit of detection that are absolutely acceptable in a clinical context. For this reason, I want to mention that there's a number now of assays out there in the market that we are typically using in our routine activity in the context of oncology research, and some of these assays are here listed. We are using many of them in our activity. I'm referring to the Oncomine family assay, for instance, like the lung, breast, and colon cell-free DNA assay. But of course, today we will mostly focus our attention about the Oncomine pan-cancer cell-free assay that we have evaluated in our laboratory in the last couple of years. Now, once again, the portfolio that today we have as possible molecular bullets that we could use to analyze cell-free DNA is mostly referring to panels that are designed to assess mutation that are relevant for lung tumor, breast tumor, colon, and nowadays also with a broader approach, pan-cancer. Now, all those panels that you see in here are designed to detect primary tumor drivers as well as resistance mutation. They include biomarkers that have already associated label drug, or they are either included in clinical guidelines, and therefore allow also the possible enrollment of patients that are analyzed by using those tests in prospective clinical trials. Here you see some of the value when referring to analytical sensitivity and specificity for the different panels that I have listed in the previous slide. And when we're talking about the pan-cancer assay, which is the main focus of our discussion today, when a test was done by using control samples at allelic frequency and mutation at allelic frequency in the range of 0.5 down to 0.1%, sensitivity and specificity of the assay has been proven to be extremely high. Of course, it is important to mention that those parameters are very much influenced by the initial input of DNA that is used for the test. If the input of DNA of the test is reduced in the range of 1 to 5 nanogram of cell-free DNA, the limit of detection is in fact increased, while in the majority of cases when we have about 20 nanogram of starting cell-free DNA, the limit of detection of this assay is in the range of 0.1% allelic frequency, which once again I want to remark is something that it's pretty much in the range of what is needed in a clinical routine setting. Now, how did we perform the validation of some of those tests, especially of the pan-cancer assay? Of course, we collected a number of samples where we had enough material to perform orthogonal tests with different approach. We have analyzed this in between in our test 40 to 60 samples. Samples have been analyzed in parallel by using digital PCR as well as NGS, and data have been generated and concordance rate have been then assessed. So once again, the purpose was to validate workflow in a real sample setting, and the procedure was to use an orthogonal method, in this case digital PCR, to confirm the result obtained by NGS. Once again, I would like to spend a few words about validation procedure. I apologize, this slide, it's quite busy. I think you could read what is written later on, but from a graphical point of view, I would like to just quickly guide you through what I depicted in here. And the concept is that we typically start our validation of an assay by using reference sample. For example, artificial DNA, which has been constructed in order to have very specific set of mutation. Typically, we will move on after this first step into a second step of validation, which requires the usage of real sample used in a real setting and analyzed and validated by using an orthogonal method. And last but not least, we regularly check the performances of those assays as well as the performances of our laboratory into international round-robin tests, and there are several of them that are nowadays available for cell-free DNA, especially I'm referring to the one organized by the German Society of Pathology, the KIP, as well as the one organized by the MQ and other professional associations that provide very well organized international round-robin tests. Those are the typical three steps that we're going to before we introduce a new assay into our routine clinical activity. Now, as I mentioned, pan-cancer assay is a targeted NGS sequencing approach. And here I just would like to briefly mention how it works. Typically what we have in our assay is an extraction of a genomic DNA, which is mixed also with cell-free and RNA. The targeted enrichment of the pan-cancer assay allows to have more than up to 50,000 reactions in a single tube that allows specific amplification of certain genomic regions that are interesting for our analysis. And then after generating an NGS library, this allows by bioinformatic tools to spot possible variants in the genome. Those are just the most important step that you need to go through in order to generate data from cell-free DNA. And I would like to guide you step by step through the different to the workflow that we are typically using in our setting. First of all, that's collection of the blood samples, which needs to be separated in order to extract the plasma. Let me here just mention that it is important, or it is suggested to use plasma instead of serum, simply because the amount of cell-free DNA that is found in plasma is typically higher than the one that is typically found in the serum, and it has been shown in a number of different papers and international validated studies that plasma is the best option when it comes to cell-free DNA analysis. After the plasma separation, we will typically proceed with the extraction of the cell-free DNA or the RNA using a bit-based or a column-based system, and the typical yield, as I mentioned before, is between 5 and 20 nanograms per ml of plasma. After extraction of the nucleic acid, we will proceed with the library preparation using a target enrichment and the molecular barcoding technology. After library is separated, we will proceed with the sequencing of targeted region by using a sequencing instrument. In this case, what you're looking at is an S5 XL. And then we will eventually go on with data analysis and, most importantly, with variant identification and interpretation. And I will really now to stress out the point that among all those different points, by far, the most complicated one is, in fact, the last step. So data analysis and variant identification, along with variant interpretation, is indeed the most complicated part of the entire workflow. Let's start with plasma separation. Plasma separation is typically done by spinning out the blood samples 30 minutes at 1.6,000 G, and transferring this in a tube to spin it again in order to have a proper separation and to remove the cellular component from your samples. So that's what you typically ending up for, and it's in this component here that you will find the cell-free DNA or the cell-free RNA that you will need to make your analysis. After this first step, a second step of extraction needs to be performed. Here, as I mentioned before, we used a bead-based solution, which is called MaxMax cell-free DNA or RNA isolation kits. And I also want to really stress out the point that it is important that upon this process, which is actually a pretty straightforward process, you elute your sample in as small a volume as possible. What we typically do here in Basel is that we elute our samples in not more than 50 microliter of volume. This will allow you to have much more flexibility in your downstream application. But I also would like that it's extremely important to perform a quality check after the extraction of the nucleic acid from the plasma. And this quality check we typically do by using a bioanalyzer or an Agilent tape station. So therefore, looking at the profile of the extracted cell-free DNA. This is how a typical profile should look like. That's the cell-free DNA peak component. And here, you will always have in your extraction a bit more of genomic DNA carryover, but the amount of genomic DNA carryover should be definitely lower than the amount of your cell-free DNA. And if this ratio is not maintained, as the one I'm showing here, you might have some problem later on in the data analysis if your cell-free DNA is too much diluted by the genomic DNA. After cell-free DNA extraction, the next step will be generating a library by performing a target enrichment. What we're using here, and this is valid for all the Oncomine product as well as for the pan-cancer assay, we use a very specific and peculiar technology, which is based on the molecular tagging of the specific set of primers that are used to enrich our targets. Molecular barcodes, without going too much into the technical detail, it's a technology that allows you to go down to allelic frequency that are extremely low, much lower than the one that are typically used in a library generated when starting from tissue samples. And this is because molecular barcoding allows you to generate families of amplicon that can be later on analyzed in order to exclude artifacts that are associated by PCR amplification. After generating the library, this needs to be sequenced, and as I mentioned before, we use the Ion S5 system. So the library is loaded into an ion tower in a chip. We typically do this here in Basel by using the Ion Chef, and this is the output of a typical run. What you're looking at here is a 540 chip with a very good level of loading, with an acceptable level of polyclonality. I'm sorry, you're looking here at the 530 chip. I apologize. With a very good level, with a very good number of reads, with an acceptable level of polyclonality, and with the shape of the library that it's very typical depending on the panel that you're using. And this is the shape of the library that you should be looking at when you, for example, are generating a library by using the cell-free DNA Oncomine lung assay. So after the sequencing is done and the quality check is properly made on the server, you need to push those data into a software that allows you to generate, actually to make the data analysis. We typically do this by using the IO reporter, which allow us to detect and make interpretation of variants. This is a fully automated variant caller system, which has a very specific workflow for each of the different panels that we are using, and it does provide a specific panel as well for the pan-cancer assay, and it has optimized parameter for the analysis of liquid biopsy. This is the typical output that you would generate out of an IO reporter analysis. You would have the genes that have been identified, the allelic frequency of the mutation found, the amino acid change that is associated to the mutation, as well as the limit of detection calculated for your assay. Important to mention is that the assay that we're discussing today are not just able to look at single nucleotide variants, but are as well offering you the opportunity to look at copy number variants. And some of the copy number variants that we will be discussing in our results are in fact extremely relevant from a clinical point of view. Okay, so we have now analyzed the entire workflow, the typical workflow, and the different steps that are that you need to work through in order to generate your data. And now I would like to present a bit more in detail the Oncomine pan-cancer-free assay. This assay allows you at the very same time to analyze both DNA and RNA without having any specific addition to your protocol. It allows to tackle variants both in this both type of nucleic acid. The pan-cancer assay has 52 unique genes. 50 of them are covered in the DNA part, 12 of them are covered in the RNA part, some of them are clearly overlapping. And you see that those are the hotspot, the genes where we have hotspot mutated covered. Those are some of the tumor suppressor genes. Those are all the genes that you could actually assess copy number variant. And let me point out that among some of these, there's LBB2, also known, of course, as HER2, MET, as well as the FGFR family. And very importantly and clinically relevant, with this assay, we can also look at gene fusion, such as fusion in the ALK, RET, ROS, and NTK 1 and 3 genes, which once again are extremely relevant for a clinical setting. The panel comes with a single pool design. In terminal specification, it allows you to go down to allelic frequency down to 0.1% when starting with 20 nanogram of input. Copy number alteration can be detected as far as those are have a fold change higher than 1.4. And fusion can be detected if they are present at least in 1% of the reads based on the analysis of the RNA. Important again to mention is that those genes have been selected based on relevant evidence that have been obtained by looking at guidelines and labels derived from Food and Drug Administration, EMEA, and guidelines from NCCN, ESMO, as well as global clinical trial that you can find at the NCI database. Now, important to mention is that the pan-cancer assay will allow you to analyze variants that are relevant for a very extensive number of tumors. And here you see the top pan-cancer cell-free DNA genes ranked by mutation for a number of different tumors that you could actually analyze by using this panel. As you can see, there's quite consistent number of genes included in the panel that is typically mutated in all these different kind of tumors. This allows quite an extensive level of flexibility to use this panel in very different settings. And here now some of the data that we generated here in Basel. What we did, in order to have some kind of reference, is to compare the performances of the Oncomine cell-free DNA lung assay that we are now using since more than 1 year in our laboratory, compared to the one that we obtained by using in the Oncomine pan-cancer assay. First of all, let me start from this part of the slide, which you can find in the top left side. Last year, I just pulled out the samples that we analyzed in the last 12 months. And in the last 12 months, we have analyzed 93 liquid biopsy using the Oncomine cell-free DNA assay. And for a vast majority of them, we have been able to obtain the sequence. So the very few samples, only 5 samples out of 93, where we did manage to obtain sequencing. And the reason for this failure can be mostly found in the insufficient amount of cell-free DNA. Now, if you compare this result to the one that we obtained by using the Oncomine pan-cancer assay, you have a very similar situation. Indeed, out of the 22 analyzed samples with the pan-cancer assay, there has been only one so far where we couldn't generate a usable library. Indeed, both the assay have a very, very high level of successfulness when generating the library. Now, the second parameter I would like to focus on, once you generate the library, it doesn't mean at all that it can be useful, can pass your QC in order to make a proper analysis. Indeed, an additional step needs to be done, as the one we quickly mentioned before in my previous slide. And indeed, once again, the performance of both assays is relatively satisfying. For the cell-free DNA lung, the vast majority of what we have analyzed has passed our QC test, and the same is true for the Oncomine pan-cancer, where in fact only 1 sample out of the sequence one didn't pass our QC, meaning that we were not able to generate enough reads, we were not able to generate good quality reads, but in fact, for the vast majority of our samples, more than 95% of them, we have been able to generate very solid data that allow us to make very consistent and solid analysis downstream. The other thing that I would like to focus your attention on is this graph on the right side of the screen, where you see that there's almost, although not a linear, but almost a linear correlation between the input of DNA that you are putting into the assay and the output in terms of library. And in red, you see what we obtained by the pan-cancer, and in blue, you see what you have obtained by using the cell-free DNA lung assay. Another thing that I would like to mention is that when we made a direct comparison between the result obtained by the Oncomine cell-free DNA lung assay and the one obtained by the pan-cancer, we actually obtained 100% concordant rates in the 10 analyzed samples with both assays. And when we were looking at limit of detection at 5%, 1%, and 0.1%, both assays have a very, very high level of sensitivity and specificity, performing very well also on extremely low level of detection. I also would like to point out here that in order to obtain very high quality data and in order to make downstream a very consistent and solid analysis, I strongly advise you for the pan-cancer assay to obtain something in between 20 and 25 million reads out of your sequencing. Now, the other thing that I really would like to point out, and I'm getting to the end of my talk now, is that there's a number of additional possible applications for the cell-free DNA that still needs to be validated, where we have different level of evidence in the clinic. First one is screening and early detection of cancer. I have to say that there are at the moment very low evidence there that we could really use liquid biopsy for this application, but data are piling up. Stratification and therapeutic intervention is a very interesting point at where we are gaining a lot of data at the moment. Real-time monitoring of therapy, once again, there's a medium level of evidence for using liquid biopsy. Risk of metastatic relapse, unfortunately, this will be extremely interesting, but there's very low level of clinical evidence for using liquid biopsy in this case. But what is really striking here is that therapeutic guidance and resistance mechanism understanding and identification of mutation is something that nowadays has an extremely high level of evidence, and this is in fact the most used application for liquid biopsy in routine clinical setting. Now, for my conclusion, I would like to mention that despite tissue biopsy still remains essential, at least for the primary diagnoses, given the fact that it gives you information about morphology, tumor type, site of origin, and immunomicroenvironment, liquid biopsy is really now rising up as a reality, and in the near-to-come future will definitely make its ways to the clinical setting. However, vigilance and attention must be paid in interpretation of results before those results can be firmly established into a daily clinical practice. For several applications, this has been already achieved, and again, I'm referring to resistance mutation monitoring, and I believe that the Oncomine pan-cancer assay offer really the possibility to analyze a large number of tumor types with a very technically robust background in terms of reproducibility, specificity, and sensitivity, which will definitely offer us a very good solution in our routine setting. And in the future, additional studies are required in order to assess clinical validity and utility for cell-free DNA analysis, which we have only partially touched upon today. With this, I would like to conclude once again with a disclosure for me for this talk. And most importantly, I would like to acknowledge all the people working in my team. They did a fantastic job in evaluating this panel. I would like to thank all the people working in the R&D unit here in Basel, as well as the lung specialists, the molecular pathologists, and all the people working in the lab. This is purely a dream team, and I would like again to conclude by saying technology is extremely important, but also people, well-trained people, are as important as technology to be able to achieve the best result. And with this, I would be happy to take any of your questions.
Thank you, Dr. Quagliata. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. Before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar. And for those who'd like to continue with our three-part New Frontiers in Liquid Biopsy Research series, Part 3 will take place on May 22nd. We will now start the Q&A. If we want to detect mutation from liquid biopsies at early stages, what kind of tumor marker is expected to show up first? Is it ctDNA, CTCs, exosomes, or something else?
So, thank you for the question, first of all. As I also stated in my talk several times, at the moment we really using only cell-free DNA and for a limited number of applications. The other one are certainly very interesting, but there is not enough clinical evidence for using them in a routine setting. Indeed, I would be extremely interested to use CTC, for example, because from them you can get not just nucleic acid, but also protein, and therefore you could also look at expression level of certain genes, but at the moment this is not something that is used in a clinical routine setting.
Luca, what opportunity does liquid biopsy testing present for early cancer detection?
Well, there's been a lot of discussion about this, I have to say, and once again, this is a very good idea. There is a lot of hope in this, but unfortunately at the moment, I have to say that data are missing to support this application. In fact, the major problem in detecting early stage of disease is the fact that early stage cancer have shown to have a very limited number of shedding a very limited amount of cell-free DNA into the circulation. That's why nowadays, with the technical sensitivity that we have at the moment, it's going to be extremely challenging to detect such kind of a mutation. A number of papers, although a very limited number of people, has shown some of these data. And the major problem I personally have at the moment is that sensitivity and specificity of the tests that have been used in this manuscript is very limited. In fact, the major problem is that when you have a mutation, you can clearly say, okay, this is coming from a tumor cells in the vast majority of cases. The problem is the interpretation of negative result. When you have a negative result, you are not actually in the position of saying that there is in fact no tumor. And this represents at the moment a major problem.
How would you address sensitivity and specificity for liquid biopsy samples from patients who have undergone organ, tissue, or bone marrow transplants?
That's a very, very good question, I have to say. At the moment, we don't have, I would say, a very well-defined and, I would say, properly designed strategy in order to assess those parameters in this very specific setting. And I have to say that indeed, in fact, for this very specific application of patients that underwent bone marrow transplantation, we are not using currently liquid biopsy. That's why, unfortunately, I cannot give an appropriate answer in this case. I apologize.
And going back to tumor mutation burden, can you comment on tumor mutation burden in liquid biopsy, where the field is on that right now?
Thank you very much. This is a very good question. Indeed, tumor mutational burden has been an extremely hot topic at the moment, considering the whole attention that immune oncology has recently raised in the field. But I have to say that I've seen very limited data in terms of tumor mutational burden on cell-free DNA. And once again, the major problem is limitation of the input of tumor DNA that we can use. There have been some results, preliminary results, shown by performing an whole exome sequencing of cell-free DNA. I have to say that the overall quality of the data is limited by the fact that the sensitivity of this approach with cell-free DNA is as well very limited. So personally, I definitely foresee in the near to come future the possibility to assess tumor mutational burden in cell-free DNA, but at the moment we don't have yet an appropriate test to assess TMB in cell-free DNA.
What extraction method or kit did you use for the samples used for validation?
As I mentioned, mostly two types of approaches you can use, column-based approach and bead-based approach. In our validation, we actually use a bead-based approach because in our hands it has shown to actually come back with the highest yield of cell-free DNA extraction. But I want to mention, most importantly, it's not just about the yield, but it's about being able to extract the appropriate fraction of DNA. And that's exactly what we want to have. We don't want to have that much of genomic DNA in our extraction because this will actually dilute and change the ratio versus the cell-free DNA. And that's why so far the bead-based method have shown to be so far the best at the moment.
Are there cancer biomarkers that are perhaps not gene-based that are circulating in blood at higher concentrations than cfDNA?
I apologize. Could you just quickly repeat the question? I'm sorry about that.
Certainly. Are there cancer biomarkers that are perhaps not gene-based that are circulating in blood at higher concentrations than cfDNA?
I have a problem, unfortunately. Again, I apologize with the connection. I'm really sorry. Could you repeat once again for the third time? I apologize, all participants, for this.
No problem. Can you hear me now?
Yeah, now I can hear you. Sorry. Yes.
Okay. No problem. Are there cancer biomarkers that are perhaps not gene-based that are circulating in blood at higher concentrations than cfDNA?
Oh, yes, that's a very good question. Thank you very much for asking. Yeah, there are for sure a lot of other biomarkers other than cell-free DNA that are being currently used also in the clinics, and I'm referring to CA, I'm referring to PSA as well, and other of these biomarkers that are in fact found at highest concentration compared to cell-free DNA in patient blood. But that's one major problem. The problem is always sensitivity and specificity, and also prediction value of those biomarkers. What I'm referring to is the fact that typically those biomarkers can, the level of those biomarkers can increase in many different conditions. For example, if you refer to a very practical example, which is CA 125 in breast cancer, which is typically used in breast cancer patients, these biomarkers, which can be assessed in the blood of the patient, can also increase in non-tumor related conditions like very strong and very enhanced level of inflammation, which are not exclusively related to cancer. And this is the major problem that we have. Differently, cell-free DNA, and especially tumor DNA, it's something that can be most exclusively generated by tumor cells, and that's why this allows us to have a higher level of robustness and also consciousness in the analysis that we are doing. I would definitely say it's not the only way to go, but in association with other biomarkers could actually increase the overall sensitivity and specificity of the entire diagnostic procedure.
Great, thank you very much, and that's all the time we have for today. We'd like to thank Luca Quagliata and our sponsor, Thermo Fisher. If we didn't have time to get to your question, we will try to follow up with our expert. As a reminder, please look out for our pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.